Welcome students. Assalamu alaikum wa rahmatullahi wa barakatuh. Today I am going to talk on one of the important chapters in your syllabus, that is classification of living organisms, proposed by R.H. Whitaker in 1969. According to Whitaker, living organisms are divided into 5 major kingdoms. First kingdom is kingdom Monera, second is kingdom Protista, third one is kingdom Fungi, fourth one is kingdom Botany, and the last one is kingdom Animalia. Today we are discussing about first kingdom, that is kingdom Monera. That's also called as bacterial kingdom. This kingdom includes ancient prokaryotic unicellular organisms, and in this kingdom we have to study about bacteria, which are divided into 2 types. One is eubacteria, also called as true bacteria, and second one is archaebacteria or ancient bacteria. This kingdom also includes organisms like mycoplasma, rickettsias, etc. First, I will tell about kingdom Monera. Kingdom Monera is also called as prokaryotic unicellular kingdom. It's also called as prokaryotic unicellular kingdom. This kingdom includes ancient, primitive, prokaryotic, unicellular organisms. Like you have first category, that is bacteria, which are divided into 2 categories. One is called as eubacteria or true bacteria, and second one is cyanobacteria, also called as blue-green algae. And this kingdom also includes organisms like archaebacteria, archaebacteria. Like you have thermoacidophilus, mechanogens, or halophiles also. And this kingdom also includes organisms like mycoplasma, earlier called as PPLO abstract S, that is Pleuro, PLEURO, pneumonia, PNEUMONIA, Pleuro pneumonia like organisms. and sometimes rickettsias also rickettsias were also included in this kingdom Monera. Now first we will come to our first topic in your syllabus, that is bacteria or true bacteria. The word bacteria has been derived from a Latin word that is bact, B-A-C-T, bact, which in English means rod. Since most of the bacteria are rod shaped, that's why the name is given bact bacteria. First word I will tell you about the discovery of bacteria. Discovery about bacteria. Bacteria was first discovered by a Dutch naturalist. Was first discovered by a Dutch naturalist who is from Netherlands. Anthony van Leeuwenhoek, Leeuwenhoek, in 1676. Bacteria were discovered by Anthony van Leeuwenhoek in 1676. Is the same scientist who invented first compound microscope in 17th century, and with the help of his self-designed compound microscope, Anthony van Leeuwenhoek discovered RBCs, protozoans, as well as bacteria with the help of his self-designed compound microscope. And Anthony Leeuwenhoek, he observed small tiny organisms having motility, they show movement. Since these organisms are small and show motility, he called them as animalcules. Animalcules means little animals having motility. That means Anthony van Leeuwenhoek discovered these bacteria and named them as animalcules. It was subsequently Ehrenberg, Ehrenberg, Ehrenberg in 1838, who renamed these animalcules as bacteria. Therefore, bacteria was discovered by Anthony van Leeuwenhoek and was named by Ehrenberg. Then it was Louis Pasteur. It was Louis Pasteur in 1876 who carried extensive study about bacteria. Who carried extensive study about these bacteria, and he also gave a technique that's called pasteurization. And Louis Pasteur also gave a technique how to culture these bacteria, how to grow these bacteria in laboratory conditions, and how we can sterilize. He also gave the technique of sterilization. And it's also Louis Pasteur who also Louis Pasteur who carried extensive studies about rabies disease and found the cause of disease, that is rabies virus, and also developed a vaccine for this that's called anti-rabies vaccine. Anti-rabies vaccine. This rabies virus is present in the saliva of rabid dogs. Then the branch of biology or science that deals with the study of bacteria is called bacteriology. And Robert Koch is remembered as father of bacteriology. This was about the introduction about bacteria and their discovery. It is subsequently followed by another question, that is bacterial occurrence. Where bacteria are found and in which habitats, in which environment bacteria are usually found. Bacterial occurrence. As you already know that if you take a handful of soil, it contains thousands of bacteria. That means bacteria are found everywhere. They are found everywhere. That means their distribution is ubiquitous. Ubiquitous means worldwide. And bacteria are found in both soil, air, as well as water. However, some bacteria, they are found in extreme environments. They are found in extreme habitats. Some bacteria are found in extreme habitats like hot springs, deserts, or snow. These are extreme environments where bacteria can withstand, they can survive in these extreme habitats. Like you have bacteria that's called as Thermus aquaticus, THERMUS aquaticus. Thermus aquaticus is a bacteria that is found in hot springs where the temperature can rise up to 78 degrees centigrade or above. And from this bacteria, recently an enzyme was derived, that's called as Taq polymerase. And this enzyme, since this enzyme is stable even at high temperature, that's why this enzyme is used in PCR, polymerase chain reaction, in order to amplify genes, in order to amplify DNA, in order to amplify the quantity of DNA. And in this PCR cycling, we use an enzyme that is called Taq polymerase that is derived from this Thermus aquaticus, which is found in hot springs. Since we know that all enzymes are, it's an enzyme means it's a molecule which can accelerate, it's a protein which can accelerate the rate of reaction. Since we know that all enzymes are proteins, and proteins are broken down at a temperature above 40 degrees centigrade. But this enzyme, that is Taq polymerase, they remain stable even at temperature 78 or above. That's why this enzyme is very important for molecular biology, especially PCR cycling or DNA amplification or gene amplification. And this is the enzyme that's extracted from this bacteria. And some bacteria also grow on deserts, and some also survive on snow. Like you have bacteria, we call them as psychophilic bacteria. These are those bacteria which can survive ultra low temperature of minus 196 degrees centigrade. Is the temperature of a liquid that's called as liquid nitrogen. That is used for cryopreservation. And these are bacteria which are found in extreme habitats. Then occurrence is followed by another topic that's called as bacterial shape. It's followed by another topic that's called shape of bacteria, or you can say bacterial shape. Now it is followed by another topic, that is bacterial shape. Shape of bacteria. On the basis of shape, bacteria are divided into four main types. Four main types. First type you have cocci, C-O-C-C-I. Second type you have bacilli, B-A-C-I-L-L-I. And third type you have spirillum. And fourth type you have, that is Vibrio. Spherical type of bacteria are called cocci or cocci, like this. And rod-shaped bacteria are called bacilli, like this. Third one, spirillum, ribbon-shaped or spiral-shaped bacteria, like this. And fourth one, that is Vibrio, comma-shaped bacteria, comma-shaped, C-O-M-M-A, comma-shaped, ribbon-shaped. Helical type. These are four major shapes of bacteria. And spherical one is further divided into subtypes. A, monococcus, like this. Second, diplococcus. Diplococcus means like this, two spheres. Third one is tetracoccus, four. And fourth one is, that is streptococcus. That's in the form of chain, like this. It's in the form of chain. It's called streptococcus. Similarly, in case of bacillus, we have different types. First is monobacillus. Single rod. Second is diplobacillus. Diplobacillus, two rods. And third is streptobacillus. When number of rods combined, it form a structure like this. In the form of chain. In the form of chain, it's called streptobacillus. For example, in case of cocci type, we have bacteria called as Streptococcus pneumoniae. Streptococcus pneumoniae, P-N-E-U-M-O-N-I-A. It's a bacteria that causes pneumonia. P-N-E-U-N-E-U-M-O-N-I-A in animals and human beings. These are different shapes of bacteria. With this we conclude today's lecture. Inshallah, I hope you understand my lecture. Inshallah, tomorrow we'll come with the next lecture. Thanks.